Hello guys, in this video I'll be explaining the whole process of meiosis. Now what is meiosis? Meiosis is a special type of cell division that produces genetically non-identical haploid cells. Now what are these haploid cells? Let's take a normal cell or else we also call it a somatic cell. A somatic cell has 46 chromosomes and they exist as pairs. So you can see we have 46 chromosomes and they are existing in pairs. So each pair is made up of one of the father's chromosome and the mother's chromosome. So in other words, we have 23 chromosomes from our father and 23 chromosomes from our mother. This is why we share the features of both our mother and our father. Now, if we take mitosis, the normal cell division, what occurs is, let's tell a normal cell, it undergoes mitosis to produce two identical daughter cells. They will have 46 chromosomes, no change in the chromosome number. Now, if we take all the cells in our body, they undergo mitosis except for one cell. So, these cells are the gametes, the sex cells, the sperms and the egg cell. Hypothetically saying, let's tell that the sperm cell doesn't undergo meiosis, they undergo mitosis. If a sperm cell undergoes mitosis, a cell undergoes mitosis and produces sperm cells, then the sperm cell will have 46 chromosomes and the egg cell will have 46 chromosomes. Now, during sexual reproduction, when the egg cell fuses with the sperm cell, we will form a zygote and it will have 92 chromosomes, and a human being can't exist if they have 92 chromosomes. They can only have 46 chromosomes. So what we do, we using meiosis, we can reduce the number of chromosomes from 46 to 23, right? So meiosis will start with a germline cell. So what is a germline cell? That is the cell that gives rise to the sperm cell and the egg cell. And the germline cell, they have 46 chromosomes and they undergo two cell divisions. Unlike mitosis, mitosis have only one cell division and it will produce two cells. Meiosis has two cell division, meiosis I and meiosis II. Okay, so they will undergo meiosis I here and produce two cells, and they will undergo meiosis II and produces 4 cells. Now the fascinating thing is all these 4 cells are not genetically identical to each other, right? And they have 23 chromosomes, so we call them haploid. If they have 46 chromosomes, we can call them diploid, right? Now they form haploid cells and they are not genetically identical to each other. Now what does this mean? This means they have different combinations of chromosomes. Let's tell this develops into sperm cell. We can call this sperm cell A, and this develops into a sperm cell, and this we can call the sperm cell B. Let's tell the sperm cell A with a different combination of chromosome binds to an egg cell. Okay, now sperm cell has how many chromosomes? 23, and the egg cell has 23. This sperm cell A with its 23 chromosome will bind to egg cell and fuse and form a zygote, and this zygote will give rise to a whole new human. But let's tell the sperm B with another different combination of chromosome binds with this egg cell. Then what happens? It will again form a zygote, but due to the variation, it will produce a different person. Now this person and this person might look similar to each other, but they are not identical to each other because of the variations. So this is why siblings look similar to each other, but they are not identical to each other. So that is the use of meiosis. So today we will learn the process of meiosis and how meiosis causes this variation. Now, even though meiosis and mitosis are different, most of the steps are similar to mitosis. So if we take meiosis I, they have 4 stages: prophase I, Metaphase 1, Anaphase 1 and Telophase 1. Now, simultaneously with telophase, something called cytokinesis also occur. Now, if you have learned mitosis, you already know what this cytokinesis is. So, cytokinesis is the division of the cytoplasm of the two cells to produce cytoplasm of the single cell to produce two cells. So, these two occurs simultaneously. Now, these are all the stages that are present in meiosis 1. But like mitosis, even in meiosis, something occurs before it. What is that? That is the interphase stage. Very, very important, right? We have learned in mitosis that most of the cells are in the interphase stage. About 90% of the cells are in the interphase stage. Now in the interphase stage, what happens? We know the cell will grow in size, the organelle content will multiply, and also DNA replication occurs. So this is one of the most important things that occurs in interphase. What? In the S phase of the interphase, DNA replication occurs. So DNA is copied. Now all the DNA that is present in all these chromosomes, the mother's chromosome, the father's chromosome, all the DNA here are copied. Now when they are copied, how are they going to look like? Let's take only one chromosome set. Okay, another thing. Now this pair, we call it the homologous chromosomes. Why? Because each pair has the same set of genes. So let's take this pair, okay. So I am taking the first two chromosomes. If you take this pair, okay, so this is that pair. These are homologous chromosomes, which means they have the same set of genes. I'll explain what does that means. So for now, remember homologous chromosome. Let's tell this chromosome has a gene for the eye color. This chromosome will also have a gene for the eye color at the exact same location or the locus. So let's mark it. So here this chromosome has a gene for the eye color, and this chromosome will also have a gene for the eye color because they are homologs. In other words, homologous chromosomes. Again, let's tell for the hair color. This chromosome has a gene for hair color. This chromosome will also have gene for the hair color at the same exact location. Okay, but here is the twist. Even though they contain the same set of genes, they have variations, which means, let's tell this is for the eye color, right? This gene will produce black eye and this gene will produce blue eye color. So this is what we call alleles. The alterations of the genes, we call them alleles. Even though they are the same set of genes, they have different alleles. So this gene has blue allele and this gene has black allele. Same here, this gene might have black hair color, this gene might have brown hair color. So that is what alleles are, and that is really important when it comes to meiosis, and you will know why in some times. So now these homologous chromosomes, as I told, in interphase DNA is copied. Now all the DNA that is present in these chromosomes is copied. So they are going to look like this. So all the DNA in this chromosome is also replicated. So they are going to look like this. Okay. Now this newly formed DNA have the same exact DNA as this chromosome, and same here, the newly formed DNA has the same exact chromosome as this newly formed DNA has the same exact DNA as this chromosome. There is no change. Even the alleles are the same because this is a genetical identical copy of this, and this is a genetical identical copy of this, right? Now, this occurs between all the chromosomes that are present inside the nucleus. But for the sake of simplicity, I am only using two chromosomes, the homologous chromosome, a pair. So, this pair, as I told, this is from the father and this is from the mother. Okay, now let's go through prophase. But a few things before I start what occurs in prophase is, if you take these arms, right, if you take this chromosome, uh, it has two arms, one arm here and the other arm here. We refer to it as the sister chromatids. So this chromosome has two sister chromatids. And this chromosome also has two sister chromatids. Now, why are they sister chromatids? Because they have the same exact DNA material. Okay. Now, in a single homologous chromosome, we will have four sister chromatids, four sister chromatids. Now, these sister chromatids, they are held together by a protein. This protein, we call it cohesin. So, this is a ring-like protein that holds the sister chromatids along their length. So, this ring-like protein is present everywhere in these gaps, and it is highly concentrated in this middle region. This middle region, we call it the centromere. Okay. So, what does this cohesin does is, it holds the two sister chromatids together, and also cohesin is also present between these two sister chromatids. Okay, so it will hold these two sister chromatids also together. Now let's go through the prophase stage. So first thing that occurs in prophase, so let's draw a cell, right? So this is just the DNA material, the chromosomes and the sister chromatids are present. So I am going to draw a cell. So this is the cell. Now these chromosomes are present inside the nucleus, right? How many chromosomes are present? 46 chromosomes are present inside the nucleus. And how many sister chromatids are present? Totally 92 chromatids. Why? Because each chromosome have two sister chromatids, right? So 46 chromosomes will have 92 sister chromatids. Now what happens is, in prophase, the first thing that occurs is the breakdown of the nuclear envelope. So nucleus, so the chromosomes are present inside the nucleus, so the nucleus is broken down. Now in the interphase, as I told, interphase occurs before meiosis. In the interphase, some structure called centrosome was duplicated. So we have learned about the centrosomes in the mitosis video. Right? These centrosomes are duplicated and they are near the nucleus. Now the nuclear envelope has broken down, the centrosome starts moving towards the opposite poles. Okay, they start moving towards the opposite pole and they end up on this region, on the opposite poles. So I am gonna draw that. So the centrosomes start ending up here. Okay, and they start releasing something called the microtubules. So we will see the function of the microtubules in a while, and before that we have to go through something else. Now as I told, these two are sister chromatids and these two are sister chromatids. Are these two sister chromatids? So this is sister chromatid and this sister chromatid, this chromatid and this chromatid, are they sister chromatids? No. Why? Because they don't have the same exact DNA material. Why? Because they have alterations of genes. Because of that, they have slightly different DNA sequences. So we call these two non-sister chromatids. Non-sister chromatids. Now something fascinating occurs between the two non-sister chromatids, and that we call, so before that something else occurs, that is the formation of the synaptomal complex. So synaptomal complex, it's like a glue, it will hold these two chromosomes together, right, not allowing them to move away. So this is how the synaptomal complex will look like. So the synaptomal complex, as I told, will uh, bind these two chromosomes together without allowing them to move away. So this synaptomal complex, forming of it, we call the synapses. The process is called synapses. Now what happens after this synaptomal complex is formed is, these two non-sister chromatids, they start exchanging their DNA material. Now how are they going to do this? Okay, let's see. So for now I am erasing this synaptomal complex, but it is present between the cells. It is the protein complex that is holding these two chromosomes together. Now what happens is, they start crossing over. So this process is called crossing over. So in crossing over, what happens is, so the DNA, the chromosome, non-sister chromatid moves to moves to the other side, forming a egg shape. So now this sister chromatid, what happens? They start moving here. Again the same thing occurs on the top also. They will form another crossover point. Now you can see they have crossed over each other. By this, what they can do is, they can exchange their DNA material. In other words, what they are actually doing is, they are changing their alleles. We know both of these have different alleles, right? But at certain point, what are they doing now? They are exchanging their alleles, causing a recombination. Now if you take this crossing over, it can occur at minimum even 3 places in the between two chromosomes. But even in some cells, they even form in 10 places. There is a possibility for that also. But here we are only doing a crossover between two places, on the top and on the bottom. Now the places the sister chromatids, the non-sister chromatids has crossed over, right, this region we call it chiasmata. Singularly, we can also call it chiasma. Right, now we have two chiasmata here. Now what happens is, can you remember the synaptomal complex that was holding these two chromosomes together? Now they disassemble. Now we don't have the synaptomal complex. Now what happens if the synaptomal complex is not there? These cells will start to move away from each other. But they won't do that. Why is that? Because when crossing over, right, this region, the chiasmata region, they are held together by the same protein that holds the two sister chromatids together. So this is also held together by cohesin protein. Okay, now they have crossed over. Now what happens? Now can you remember the microtubules? They start moving towards the centromere, the middle region. So here also they start moving towards the centromere. Now we know, we have learned in mitosis that in the centromere we have a protein complex called kinetochore. Each sister chromatid has its own kinetochore protein. So kinetochore protein is usually present here. We have two kinetochore proteins for two sister chromatids. So here also two kinetochores are present. Now what happens during meiosis is that these two kinetochores, they fuse together and they form one single kinetochore. Again same thing occurs here. They form one single kinetochore. Now these microtubules, they come and bind to this kinetochore. When they bind to the kinetochore, we call them kinetochore microtubules. Again from this region also binds to the kinetochore, we call them kinetochore microtubules. Now these microtubules, when they bind to the kinetochore, what happens is they start pulling this chromosome towards their side. So this microtubule will start moving their chromosomes to their side. Now this chromosome is starting to move this cell to this side, and this microtubule is starting to move this chromosome this side, and this microtubule is starting to move this chromosome to their side. But they are not separated. Why? Because of this chiasmata. The chiasmata has cohesin proteins that holds the two chromosomes together. So what happens is, at a certain point, they start moving backwards, forwards, and end up in the middle region of the cell. This middle region is called the metaphase plate. So this is where the prophase I is over. Okay, so let's mark it. Now what happens? Now metaphase starts. Now as I told, we have only took 2 chromosomes here, but we have 46 chromosomes here. So what happens during metaphase is, all the 46 chromosomes, they start arranging on this metaphase plate. So as I told, the middle region, we call it the metaphase plate. Now after all the chromosomes are arranged at the metaphase plate, now here we have another thing to learn, that is what we call the independent assortment. Now what is this independent assortment? Now what is the final outcome of this meiosis I cell division is to produce 2 cells, right? So we started with 1 cell, and after meiosis I we are going to produce 2 cells. Now what happens is, this chromosome with their sister chromatid can move to the left side, and this chromosome with their sister chromatid can move to the right side when the when the cohesin in the chiasmata is broken, which is going to occur in anaphase. So these can move away from each other. The fascinating thing is, now this chromosome is on the left side and this chromosome is on the right side. Independent assortment means this chromosome can be on the left side and this can be on the right side. They can exchange their places. Now if a single chromosome exchange their places, we have 2 possible cells, we have 2 possibilities. If a single chromosome, right, so this is one single homologous chromosome, if they change places, we will have 2 possibilities, right? What are the 2 possibilities? This cell can go to the right side, this cell can go to the left side. That is one possibility. This cell can go to the right side and this cell can go to the left side. That is the second possibility. Now if we take all the 46 chromosomes, what are going to be the possible combinations? So this is for one homologous pair. For 23 homologous pair, we can have 8 million combinations. Fascinating, right? Think about it, 8 million combinations, because you don't know which homologous pair will go to which side, and we have 23 pairs. So this is what we call the independent assortment. Now what does independent assortment do? It will increase the variation. So that is our main goal in meiosis. Now we have increased variation in 2 places. Remember, we have increased variation in independent assortment, right? And we also have increased variation by forming recombinant chromatid, right? So they will form recombinant chromatid. How they will exchange their DNA material. So we will see during anaphase how they are going to look after they exchange their DNA material. As I told, in metaphase, all the homologs, they align in the metaphase plate. Now let's go through what occurs in prophase. Before that, okay. Now what happens during crossover? So this is how our new chromosomes are going to look like. So this is the maternal chromosome, right? The father's. So this is how it was originally. And this was our paternal chromosome with its sister chromatid. And this is how it was originally. Right? When we started the process of meiosis, this is how they looked like. Now, after crossing over their regions, how are they going to look like? They are going to look like this. Sorry, I'll draw it in blue. So this region, so this chromosome will gain a part of this chromosome. So they are going to look like this. They have gained a new region. Now this we call a recombinant chromatid. The same occurs in the next chromatid, the maternal chromatid or mother's chromosome. These markers. They start exchanging their DNA material, right? Now can you see they have exchanged their DNA material. So this is an unaltered cystochromatid. They have not got recombination, and this is an unaltered cystochromatid. But can you see these two cystochromatins? They have altered, they have undergone recombination. They have formed new regions from this this cystochromatid. So this part of this cystochromatid is here now. Part of this cystochromatid is here now. Cystochromatid is here now. Now, what does this cause? This causes more variations in the chromosomes. Now let's go through anaphase. Now at the start of anaphase, what happens is this cohesin that is present in between the arms of the chromatids, they are broken down. Now when these cohesins are broken down, what happens? Cohesins that are present in this region, right? Cohesins that are holding the crossover regions together, the chiasmata together, they also break down. So now the two chromosomes can move away from each other. Now this is how they are going to look after they move over. I feel so good erasing this. No, it was all like a mess. Okay. So now we have the centrosomes, right, on the two opposite poles. What happens? The chromosome with their sister chromatid move away from each other. So they are going to look like this. So this is the kinetochromatoid that is pulling them. So they are going to look like this. First, let's draw this chromosome. Right, this is the unaltered region, and this is the altered region. So now the altered region, they are going to look like this, right? Because they have exchanged their DNA material. Now the same thing occurs on the other side. Sorry, I think I should draw it in blue. So they have a region that they have exchanged their chromosome. Right? So that is this region, exchange their genetic material. Now they are moving away from each other towards their pole. Now as I told, only certain microtubules are binding to the kinetochore and pulling them. What about other microtubules? So other microtubules, we call them non-kinetochore microtubules. These non-kinetochore microtubules, what they do is they interact with each other using certain motor proteins, and they start to make the cell bigger. How? These two non-kinetochore microtubules, they pull away from each other. When they pull away from each other, centrosome moves back and the cell becomes bigger. So now this is what occurs in anaphase. So now metaphase is completed, anaphase is also completed. Now telophase and cytokinesis occurs simultaneously. Now what occurs in telophase? So in telophase, these microtubules, they uh, break down, they disassemble, right? So this is how it's going to look. The nucleus will reform. Now inside a single cell we have two nucleus, and these cells have moved away from each other. Nucleus has moved away from each other. So nucleus reforms, they have the centrosomes in the corner. Microtubules of centrosomes has broken down. Now cytokinesis takes place and the cleavage furrow forms, right? What happens? The cleavage furrow will break down the cell into two. Now we'll have two cells, right? Now you can see we have two cells and the sister chromatids with their chromosome has moved away from each other. Now in a single cell we have how many chromosomes? So let's tell we had 46 chromosomes, right? 46 chromosomes and 92 sister chromatids initially. When they divide into two, we'll have 23 chromosomes on one side. So we'll have 23 chromosomes and 46 sister chromatids, right? Each chromosome has two sister chromatids. So this one chromosome has two sister chromatids. So 23 chromosomes will have 46 sister chromatids. Now the same thing occurs here. They also will have 23 chromosomes and 46 sister chromatids. Now at the end of meiosis 1 we have produced two new cells. Right, now we can go into meiosis 2. Okay guys, now let's go through meiosis 2. Right, we completed meiosis 1 and we produce two cells, right? Two cells with 23 chromosomes and 46 chromatids. Now our homologous chromosomes have moved away from each other. Now here you can see we have an unaltered sister chromatid and we have a recombinant sister chromatid that has gained DNA regions from this chromosome. And again the same thing here. This is an unaltered chromatid and this is a recombinant chromatid, which means they have gained certain regions from this chromosome. Now meiosis 2 is really easy. Meiosis 2 is really similar to mitosis. So if you take meiosis 2, right? Okay, let's write it down here. They have four stages. They have prophase, phase 2, metaphase 2, Anaphase 2 and Telophase 2. And they have cytokinesis, right? Okay. Now what happens is, as usual in prophase, the nuclear envelope breaks down. We don't have the nuclear envelope. Now these two centrosomes, they start moving away from each other towards the two poles. So they are going to end up here. And as they are moving towards the pole, they start releasing microtubules, right? And they also start releasing microtubules radially outwards. We call this asters. So centrosome, that's the same thing. Yeah. Okay. Now we don't have the nuclear envelope. Now what happens is, so this is the middle region of the two sister chromatids. We call it the centromere, and there we have a protein complex called kinetochore, right? On both of the sides we have two kinetochores, and we saw in meiosis 1 these kinetochores, they fuse together, but in meiosis 2 they are not fusing together. Now what happens is these microtubules come and bind to this kinetochore, and they also start going towards the other pole. So when the microtubules bind to kinetochores, we call it kinetochore microtubules. Now this kinetochore microtubules, what they do, they start pulling the chromosomes the sister chromatids towards their side. But they are held together by cohesins in the middle. Along the length of the sister chromatids, they have ring-like proteins, as I told you guys. They are called cohesins, and they hold the two sister chromatids together. So what happens? A tug of war is happening right here. So this microtubule, kinetochore microtubules, is pulling this sister chromatids towards their side, and this kinetochore microtubule is pulling sister chromatid towards their side. Now, so they are moving side on this side, that side. At one point, they start aligning on the middle part. So this is when metaphase starts. So this is what we call the metaphase plate. Now the prophase is over. Right? What occurred in prophase? The centrosomes move towards the opposite side. Centrosome starts releasing microtubules. Microtubules bind kinetochores, producing kinetochore microtubules. At kinetochore microtubules, what they try to do? They try to move the sister chromatids toward their side. At one point, what happens? Sister chromatids, they start lining up along the middle line, the equator. We call it the metaphase. Now what occurs during metaphase 2 is same thing. So same thing occurs here, right? The centrosomes move towards the opposite side. They start binding to kinetochore. Kinetochore microtubules start pulling each other up to their side, and at one point they are going to end up in the middle line. Now all the 23 chromosomes that are present inside this cell is going through this process. Now what occurs in metaphase is all the chromosomes line up along the middle line, along the metaphase plate. So this is what occurs in the metaphase 2. Now what happens at the end of metaphase 2, these cohesin molecules are cut using a certain enzyme. When these cohesin molecules are cut using a certain enzyme, then what? The sister chromatids can move away from each other. When they move away from each other, they are going to look like this. So we will have the unaltered sister chromatid right here, and we will have the other sister chromatid. Now this sister chromatid, what's the difference? They have a region from here due to recombination, right? I have been telling it now for like 10 to 12 times. Now same thing occurs here, right? They start moving away from each other. Okay, they start moving away from each other towards their pole, right? Then what happens? Then telophase takes place. Now after all the sister chromatids start separating and moving towards a pole, then what happens? We will have 23 sister chromatids on this pole and 23 sister chromatids on this pole. Now the thing is, we don't call them sister chromatids anymore. We call them chromosomes because they have separated. We can call them 23 chromosomes. And here also we can call them 23 chromosomes. Now we have 23 chromosomes on this side and 23 chromosomes on this side. Now the microtubules break down. We don't have the microtubules anymore. Centromere will be there. Sorry, the centrosome will be there on the corner. Now what happens? The nucleus reforms, right? Sorry, the nuclear envelope reforms. And now as I told, for simplicity I have drawn only one chromosome. Inside this cell we have 23 different combinations. Inside this we have another 23 different combinations. Same thing occurs here. Here we will have another different 23 combinations. Here we will have another difference is the 23 combinations. Now what happens? Now cytokinesis occur. After cytokinesis, what happens? These are going to be separate cells. So after cytokinesis, this is how the nucleus is going to look like. Right? Now we can form cells around them. Right? So this is one cell, this is another cell, this is one cell, and this is another cell after cytokinesis. Now we have four cells, right? What happened, as I told at the start of meiosis, we start producing four cells which are haploid. If you take these four cells, they have 23 chromosomes only, right? So this has 23 chromosomes with a different combination. This has 23 chromosomes with a different combination. They all have variations. They are not the same. They are not genetically identical. Now each of these, what they can do, they can develop into a sperm cell or an egg cell. So these 23 chromosomes, they will be present in a sperm cell or a egg cell. So this sperm cell, they will bind to a egg cell, right? And they will fuse together and form a fertilized egg cell which will grow into a whole new organism.